So for this video here, I'm going to solve a few Hardy Weinberg problems. And so I would suggest you hitting pause in the video while you can read the question, and then I'm going to solve it. So then as you solve it on your own, then hit play and then check yourself. So here you see an allele for white wool is dominant over the allele for black wool in sheep. So in a sample of 900 sheep, 891 are white and 9 are black. Calculate the allelic frequencies within this population, assuming that the population is in Hardy Weinberg. So the allelic frequencies are asking for a P and a Q. So what I have here is I have 9 of the sheep out of the 900 are recessive phenotypes. So recessive phenotypes are Q squared. And so when I look at this, I see that 9 out of 900, that's a percent or a proportion of the population that is homozygous recessive, and that's equal to 0.01. And so when I look at what that is my Q squared, my number of individuals that are homozygous recessive. So now the question is asking to find the allelic frequencies. So when I square root this on both sides, I find that Q is equal to 0.1. Well, because P plus Q is equal to 1, if Q is 0.1, well then P would have to be 0.9. So that would be my answer. All right, next one. A survey reveals that 25% of the population of 1,000 individuals have attached earlobes that are a recessive trait. Assume the population fits the parameters of the Hardy Weinberg principle, and what is the frequency of the recessive allele? So here we're asking for an allele, so that's Q. Up here we have the percent of a population that has the homozygous recessive trait, so that's my Q squared. So what I have here is Q squared is equal to 0.25. When I take the square root of that, I find that Q is equal to 0.5 and P is equal to 0.5. All right, but really they're just asking for Q. 9% of a population is homozygous recessive at a certain locus. Assuming that the population is in Hardy Weinberg Equilibrium, what is the frequency of the recessive allele? Okay, so 9% of a population is equal to our Q squared, because that's the amount of individuals that are homozygous recessive. Then I find my Q, I take the square root of both, is 0.3. So Q is the recessive allele frequency. And that's your answer. All right. In certain Native American groups, albinism is due to a homozygous recessive condition in the biochemical pathway for melanin is sometimes seen. If the frequency of the allele, for this condition, well, albinism is homozygous recessive. So this condition is talking about that, is 0.06. What is the frequency of homozygous? Let's make it homozygous dominant individuals. So with this, okay, they straight up give us that Q, the frequency of the recessive allele, is 0.6. So P is going to be 0.94. Well, homozygous dominant individuals is P squared. So I'm going to do 0.94 squared. And that's equal to 0.88. Now, when you think about the bigger picture of this is saying that 88% of the population is homozygous dominant. And if I really wanted to find what the percent of the population that was albino, I would do 0.06 squared, and that's equal to 0.0036, or 0.36%. So less than 1% of the population is homozygous recessive. That kind of tells you something about that. Maybe it's a brand new mutation, or maybe it's selected against and has a low fitness. If you think about being albino, you don't have pigment, you don't have any natural protection to the sunlight. So that would actually explain why the dominant allele is at such high frequencies in that population. Okay, in a population of grasshoppers, the allele for tan color is dominant to the allele for green, so green is recessive. A drastic increase in rainfall leads to selection against the tan phenotype. When the rainy season ends, 23% of the remaining grasshoppers have the green phenotype. Ooh, so that's my Q squared. Green phenotype individuals, 23%. It's a 0. If this population is now in Hardy Weinberg, what will the frequency of the tan allele be in the next generation? Tan allele is P. And in the next generation, well, if it's in equilibrium, it means it's not evolving. So whatever it is now, it's going to be next year. So we need to take our square root of 0.23, take our square root of Q squared, and we'll get our Q. When we get that is equal to 0.48. So our P is equal to 0.52. And it's asking for the tan allele, so the answer is that. Cool. If 116 out of 200 individuals express the dominant phenotype, what is the frequency of the heterozygotes in the population? Cool. Well, 116 out of 200 is equal to the dominant phenotype, so that is this. So 116 out of 200 does not equal P squared. So we have to now do, well, how many are recessive? So, 116. We find that 84 are homozygous recessive or have the recessive phenotype. So now we would have 84 divided by 200. And that gives us 0.42. That is our Q squared. So when we take the square root, we find that our Q is equal to 0.65, or P is equal to 0.35. So our question's asking for heterozygotes. 2 times 0.35 times 0.65. And our overall is equal to 0.46. About 46% of the population is heterozygous. Okay, some people have the ability to taste a bitter chemical called phenothiocarbamide, PTC. The ability to taste is due to the presence of at least one dominant. So tasting is dominant. Non-tasters is recessive, 45% of the population. Assuming it's in Hardy Weinberg, what percent of the population is homozygous dominant for the ability to taste? So, okay, so we have 0.45% are non-tasters. That's our Q squared. So we take our square root of this. Let me make sure I did that right. 0.45 square root. We find that Q is equal to 0.67. And so then our P is equal to, P is equal to 0.33. And so with this homozygous dominant, you would do the, for homozygous dominant, you do the 0.33 squared. So then you would square this to get our P squared, and you get 0.11. So about 11% of the population is homozygous dominant. Okay, and here's our last one. Scientists are studying several squirrel populations on neighboring islands in the North Pacific, and the previous genetic analysis has shown that a single gene controls tail length in the squirrel population, and that the allele for long tails is dominant to the allele for short tails. On 2 separate islands, scientists recorded the number of squirrels with long tails and the number of squirrels with short tails, and here the results are shown below. So calculate the relative genotype frequencies. That means calculate these for each island. Okay, so, sorry about that. So let's go ahead and look back at this. So when we see this, our first step is to find our P and our Q for both islands. So for island 1, I'm going to take 612 divided by the total. And that's going to give me my Q squared. Oh my gosh. Q. I don't know why that keeps happening, sorry. So what we can do is we can take 612 divided by the total population. And we find our Q squared is equal to 0.27. When we take the square root, what the heck? When we take the square root, we find that our Q is equal, so I took the square root, 0.52, and our P would be 0.48. Now for the second island, we would take our recessive divided by the total. And that is equal to 0.34. We take the square root of that. We find that our Q here is equal to 0.58, and then our P is equal to 0.42. Now that you have P and Q, you would just plug them into P squared, 2PQ, and Q squared for both islands. All right, good job, guys.